Welcome to the overview video for the eukaryotic cells lesson. For this lesson, we are going to focus primarily on the structures that are found inside and outside of eukaryotic cells. Remember, that would be our plants, our fungi, our animals, and our protists. We're going to talk about the general structures. We're not going to go into structures that are uniquely found in plants or uniquely found in protists, etc. Just the general ones that are found in the majority of these types of cells. So this is the kind of typical cartoon of an animal cell. You can see, looking at it, there are a variety of different structures within the cell. It's pretty complex looking. These structures that are found within the cell are called organelles. And each organelle is basically a container that holds chemical reactions that are doing a particular thing. So each organelle has specific processes, have specific functions going on inside of them. So when you look at a eukaryotic cell, each one of these unique structures, you can think, well, what's happening there? What's the purpose of that organelle? And again, that's really the point of this lecture. I'm not going to give you a picture of a cell and ask you to, label the organelles or something like that. I want you to think about it as why. Why do you need a mitochondria? What's going on in there? Why did we evolve to have the endoplasmic reticulum? Okay. Okay, so I tend to group the organelles into kind of big classes based on function. So I think of the organelles that are involved in information movement within the cell, or the organelles that are involved with energy production within a cell, things like that. The big overall kind of arching important activities that the cell will do. So let's first dive into organelles that are involved in information. So you have hopefully done the biochemistry review or have a very strong biochemistry background, and you remember that when you look at the typical cell, you look at all of the major molecules within it, they're divided up into roughly 4 different groups: the carbohydrates, the lipids, the nucleic acids, and the proteins. So when you're talking about information, or when I'm talking about information usage within the cell, I'm primarily talking about the usage of the nucleic acids to produce the proteins that are needed for the cell to function. So remember, all of the enzymes, all the chemistry require proteins. The enzymes are proteins. They're kind of the workers in the factory, if you will. So the information is stored in a molecule called DNA. That is the instruction manual for the cell found in the nucleus. The nucleus's job is a container to protect the DNA. And it also protects a very important type of RNA in the nucleolus. But the videos will go into that in more detail, and we'll approach that again later on in the class. So you've got your instruction manual, your DNA. A section of the instruction manual is copied into a molecule called RNA. At this point, all you have are instructions. You don't actually have anything. I always say that it's like cooking. The DNA is like a cookbook. You can't eat a cookbook. It can just tell you what to do. So the DNA and the RNA, the nucleic acids of the cell, are just information holders. The actual product that you want, the actual, food that you're going to produce, is a string of amino acids that will eventually fold and become what we call a protein. That is the worker. That is the chocolate cake at the end of the recipe after you've been cooking. Okay? Okay, so in order to accomplish this kind of multi-step process, we have several structures involved. So first we have the nucleus, and that's where we're storing our DNA. That's where the RNA is produced, is inside of the nucleus. That little skinny RNA can leave the nucleus and go out to the cytosol. The cytosol is the fluid. The cytoplasm is the area that the fluid is in. So the cytosol would be kind of the fluid on the outside of the nucleus. Okay? And that's where proteins are made. The organelle, or the structure, the machine that makes a protein, is called the ribosome. Okay. So when you look for ribosomes within a cell, there are actually 2 major groups of ribosomes. They're what are called the free-floating ones that are just floating around inside of the cell. And then there are the ribosomes that are found specifically attached to a structure that butts up right next to the nucleus called the endoplasmic reticulum. When you look at the ER, there are there are parts of the ER that look like they have studs on the outside of them. Look like they have these kind of circles, these bumps sticking out of them. That part is called the rough part of the ER. And then there's a part of the ER that doesn't have those bumps, and that's called the smooth part of the ER. The rough part of the ER are ribosomes that are made there, or that are attached to the ER. So you have your free-floating ribosomes, sometimes called cytoplasmic or cytosolic ribosomes, and then you have the ribosomes that are stuck on the outside of the ER. So what's the difference between these two ribosomes? Why do we need two different groups? Well, it depends on what proteins are made there. So the proteins that are going to be produced on these free ones are proteins that are just going to be able to freely move around the cell and do whatever they need to do within the cell. The proteins that are going to be made on the ribosomes on the rough ER are proteins that are not going to be freely floating around the cell. So from the moment these proteins are made, they're actually fed inside of the ER, and they are contained within the ER. After they have been produced within the ER, there's a small amount of modification that can happen, some cutting, some adding on different groups. But when all of those modifications are finished within the ER, that little protein will leave the ER and go to the next, here's the outside of the cell here, will go to the next structure in the process, and that is called the Golgi apparatus. So we've got our Golgi. So this protein that was produced just on the ribosomes on the ER will be shuttled from the ER over to the Golgi, where it will be modified some more and sent to wherever it's supposed to go. So I say that because remember, you've got some proteins that are produced free floating on these cytoplasmic ribosomes. So the protein is produced and then is free to go do whatever it wants, wherever it's supposed to go inside of the cell. The proteins that are produced on the ribosomes found on the ER are ones that you don't want floating around inside of the cell. So they're proteins that don't function inside of the cell. So maybe after the Golgi, it will be shipped to the outside of the cell and it'll be released. Okay, that is referred to as a secreted protein, a released protein. Or it could be a protein that is going to be stuck on the outside of the cell, like a receptor. Or some of them are proteins that they are going to be used inside of the cell, but they're dangerous. We don't want them floating around inside of the cell. Some of them are digestive and destructive proteins. So when they're finished being modified within the Golgi, they'll actually be sent to wherever their final destination is within the cell, but they'll be sent in a special little bubble called a vesicle that will act like a little jail, basically, to take that protein and send it wherever it's supposed to go within the cell. So that finished product, that dangerous digestive protein, is never allowed to just float around inside of the cell. Okay, so this is one unique pathway of information flow: DNA to RNA to the ribosomes on the ER to the Golgi to either the outside of the cell or to whatever special place within the cell it's supposed to go. Most proteins are produced on the free ribosomes that are allowed, and then they just go wherever they need to go within the cell. They're not dangerous to just be free and moving around within the cell. So most proteins do not go to the ER and to the Golgi. Most proteins go to just the normal free cytosolic, cytoplasmic ribosomes that are produced that way. So I'll let you watch the videos and get more details about the smooth part of the ER, what's actually happening within the rough ER and Golgi. But I wanted to kind of put that distinction of the secretory pathway, that's what's called the ER to Golgi to released, or the free-floating ribosomes. Okay. The other thing I want to talk to you about in this lecture, or in this overview, excuse me, is the energy production part of the cell. So most people know that there are two major energy options or food options in the world. You can either make your own food like plants do, and that is photosynthesis. Oops, excuse me. Photosynthesis. Or you have to eat your food, like everybody but plants and algae, and a small group of bacteria do. Okay, so you've got the photosynthesis option, which is energy creation. So in photosynthesis, energy from the sun is harnessed into sugar. Okay, and then we eat the plant and we eat the sugar. Sugar, or carbohydrates, are the major energy source for our cells. So energy is the ability of things to do things. Okay, so it's basically the gasoline to fuel the engine of our chemistry within our cells. So how do we get energy to add, to break things or to stick things together or whatever, move things around within our body? We get that energy from our food. And the digestion of our food, the extraction of energy from food, is through a process called cellular respiration. And this is a process that you have very likely heard of before, and you probably will hear of again. It's a very, very important process. Without the process of cellular respiration, we would not be able to take any energy from our food, which means that we wouldn't be able to do any chemistry within our cells, which means we wouldn't be alive. Okay. The process of cellular respiration primarily happens in the structures called the mitochondria. So, what is food? Well, for most of us, food is sugar. It's carbohydrates. But if you never eat another carb again, will you collapse and die? No. We have other options. If you don't have enough carbs, then you go to fat. If you don't have enough fat, then you start breaking down non-essential proteins, which is why if you stop eating, you start losing muscle mass eventually. Your body starts looking for anything to break down to extract the energy out of it. But carbs, sugar, is the preferred thing. So basically, photosynthesis allows us to take the energy from the sun and convert it into sugar. And then this process of cellular respiration allows us to take that sugar and take the energy out of it, store it in a molecule called ATP. So photosynthesis happens in a structure called chloroplasts, and cellular respiration happens in the mitochondria. So this part's mitochondria, and that part is chloroplasts. So do plants need mitochondria? Yes, because they could have sugar but wouldn't be able to actually digest it and get the energy out of it. And everybody needs mitochondria. So you will watch a video all about cellular respiration in much more detail. But before I let you go, I do want to talk just a little bit more about some vocabulary words for energy. In particular, the word metabolism, which is one that we use a lot in our society. I have a slow metabolism, so I've gained weight, or people will use it, especially when talking about weight loss and weight gain. So metabolism is a word that means all of the chemistry that's happening within something. So the metabolism of a cell, or all of the chemical reactions that are happening within a cell. So that would be taking something big and breaking it down into two smaller pieces. That could be taking two smaller things and sticking them together into something big. It doesn't matter what's changing. The point is that there is some chemical reaction happening, so that is metabolism, the sum of all of the chemical reactions. And when you really look at the chemical reactions in the cell, there really truly only are two types. You're either taking something big and breaking it down, or taking smaller things and sticking them together. Even if you rearrange something, you take it off of one thing and stick it onto something else. So all metabolism, all chemistry reactions really are either making things or breaking things. The fancy name for reactions that are taking a big thing and breaking it down are called catabolic reactions. Sometimes you hear the term catabolism. And taking small things and sticking them together are called anabolic reactions. I always think catabolic is catastrophic, everything's falling apart. Or anabolic, you may have heard of anabolic steroids. Those are steroids that bodybuilders take to make their muscles build, to make them bigger. So all of the chemical reactions in the cell is all the catabolic reactions plus all of the anabolic reactions. The other important kind of concept to get here before I let you go is the concept of ATP and what it does. Well, it comes back to these catabolic and anabolic reactions. So, to make something, to take two things and stick it together, requires energy. It requires effort to take those two things and push them together and make them stick. So the attachment of these two molecules together is called a bond. That's how those two things attach together. And that physical attachment of the two things requires energy from the cells. So anabolic reactions require energy. They require effort, from the cell to make it happen. But one of the cool things about all of this is even though to stick things together is expensive, it's not energy that is completely lost. So these bonds that were created actually store some of the energy. So that when you break the bond, if you take those two things apart again, you recoup some of your costs. You receive some of that energy back. It's not 100%, it's not one for one, but you do get some energy back. So what that means is you take this big thing and break it down, you do a catabolic reaction, that releases some of that energy back into the environment. And in this case, we're talking about a cell. So you eat your Snickers bar for lunch, and it's got this lots and lots of carbohydrates and proteins and fats, which are all very, very large molecules full of bonds. And what our body does is it breaks each one of the bonds, or as many of the bonds as it can within that food. It kind of chops it all up through a process called cellular respiration. And every time it breaks one of those bonds, it releases energy, it releases energy, it releases energy. And our body takes all of that energy that's released as we're chopping up our food, and it stores all of that energy in a molecule called ATP. And the reason that we store it instead of immediately using it to do whatever we need to do in the cell is because we don't eat 24 hours a day, 7 days a week. So what happens is we eat, we break down our food, we have a bunch of bonds that we're breaking, releasing a ton of energy. Well, we use what we need at that second, but we store the extra energy in molecules called ATP. And then what happens when we need energy, we need to break a bond so that energy is released. What we do is we come up to our little ATP molecules that we made earlier when we ate, and we break that bond. So we take our ATP and reduce it down to a DP. Okay, so our ATP, we break off the third P, and we break a bond there, releasing energy, and turn it into a DP. And that energy that's released is exactly enough energy required to stick two things together, to make the typical bond. So that way, in between meals, as we need to still breathe and move and think and do all of the things that cells need to do, we have the energy source to do it. And then when you eat again and you have a bunch of food again, your body takes the energy being released and it sticks that third P back on, and it reestablishes the ATP. So this is called energy coupling. You break down one thing and use it to make something else. And ATP is the little energy kind of temporary holder. When you have lots around, you make sure to put all the P's on and make ATP, 3 P's. When you need energy, you rip one of those P's off and reduce it down to a DP, di, which means 2, a diphosphate. And then when you have energy again, you stick the P's back on, you need it, you take the P's back off. So that's what cellular respiration is all about. So you're going to watch a video that's going to go into more detail about cellular respiration. I expect you for this lecture or this lesson, excuse me, to get the general gist of energy, the gist of ATP, the general 4 phases that happen in cellular respiration. Don't worry about the details of how many ATP are produced here and there and there, just the overall big picture of the process. We'll get into it in a little bit more detail in future lessons. So, metabolism in particular can be a tough subject for people. I just encourage you for this part to keep it really big picture, 10,000 foot view. Overview of what's happening. Don't get all lost in the chemical names and NAD, 6-NADH and blah, blah, blah. No, you don't need that for this lecture. Just very big picture. Get that foundation laid, and then we'll dive into more detail later on. Good luck on this. If you have any questions at all, feel free to contact me. Post it in the Muddiest Point discussion board, and we can drill through it all together as a class and try to get your questions answered.